Welcome to the Animal Reader podcast. This week we discuss animal testing.
We often don't really stop and think about how the medicines and treatments we rely on, how they actually come about.
Right. There's this sort of common image, maybe from older films or something. Scientists, lab coats, animals.
Yeah, exactly.
Hey,
It feels like it's just accepted as the way things are done.
but how did that even start?
That's what we want to dive into today. Why animals in the first place? What was the thinking there?
Well, it actually goes way, way back. 4th to 3rd centuries BCE. You had figures like Aristotle and Erastus doing experiments on animals.
Uh-huh.
Then fast forward to 2nd century Rome, you have Galen. He's famous for dissecting pigs and goats. Later on, like 12th century Spain, there was Avenzoar, an Arabic physician. He used animals to test surgical procedures first.
Ah, okay. So practicing surgery, refining techniques.
Pretty much. More about the physical procedures.
So how did it shift towards, testing substances, medicines?
Well, over the centuries, science evolved, right?
Huh.
In the 17th century, Robert Boyle used animals for his air pressure experiments. Birds, mice.
Okay.
Then Lavoisier in the 18th century, he used a guinea pig for his work on respiration. And in the 19th century, you get Pasteur using sheep for germ theory, Pavlov and his dogs for conditioning. Animals have been part of the story. But it's important to note the formal requirement for animal testing for drugs in the U.S., that's actually more recent.
Oh.
It dates back to regulations from 1938. So while animals were used for centuries in research, making it a mandatory step for drug approval is really a 20th century thing.
Okay, got it. So historical context established. But our sources, they raise some pretty serious questions about how well these animal tests actually predict what happens in people.
Yeah, this is where things get complicated. One really striking point is about adverse drug reactions, ADRs.
Side effects, basically.
Exactly. Even when people take drugs exactly as prescribed, ADRs are a leading cause of death in the U.S.
Wow, a leading cause.
Yeah. And the sources suggest a major reason for this is that the animal tests done beforehand just aren't adequate.
Inadequate how? Like, they don't catch the problems.
Well, one study found that a massive 63% of adverse reactions seen in humans, they just didn't show up at all in the animal studies.
63%. So the animals were fine, but humans had serious problems.
That's what the data suggests. No warning sign from the animal tests for nearly 2/3 of the side effects.
That's a huge disconnect. And what about the drugs that do pass the animal tests? How do they fare later on? I remember seeing a really high failure rate number.
You're right. It's incredibly high. Our sources say around 95% of drugs that look safe and effective in animals, they end up failing in human clinical trials later. Think about that. All the resources, the time the animals used for a 19 out of 20 failure rate when it gets to people.
That's staggering. It really calls the whole predictive value into question.
It does, and it also massively slows down drug development. The average time to get a drug to market is about 10 years.
And animal testing is part of that delay.
It's a contributing factor, yeah, because you spend time on leads that ultimately go nowhere in humans.
One source called it the valley of death in biomedical science. Promising animal results just dying before they reach humans.
It's a good description. Take cancer drugs, for instance. Over 90% of those fail to help humans after showing promise in animals.
Over 90% for cancer. That's quite something. And is it getting any better? Are we improving the animal models?
Well, despite efforts to refine things, the failure rate in human trials has actually increased. It's closer to 96% now.
96. It's gone up.
Apparently so.
Huh.
And the main reasons are still the same. The drug doesn't actually work in humans, or there are unexpected safety issues the animals didn't predict.
It really points to a fundamental problem, doesn't it? That animal models just aren't replicating human diseases very well.
That seems to be the core issue. Our sources stress that animals often don't mirror human conditions accurately. The causes, the symptoms, the basic biology can be quite different.
Especially for complex human-specific diseases.
Exactly. Think about things like many psychiatric conditions, Alzheimer's. These are notoriously difficult, maybe impossible, to truly replicate in another species.
So trying to extrapolate from animals in those cases is guesswork.
One source called it an exercise in speculation, which sounds about right given the failure rates.
And we see the flip side too, right? Drugs that pass animal tests but then cause major problems in humans, leading to recalls or those serious black box warnings.
Absolutely. There are many historical examples of that. But there's also the other way around.
How so?
Think about Lipitor. Hugely successful cholesterol drug, right?
Millions take it.
Well, apparently it didn't look very promising in the early animal tests. So if they'd relied only on that animal data, we might never have gotten Lipitor. It could have been shelved.
Wow. So animal tests can fail to predict harm and they can fail to predict benefit. They can filter out good drugs too.
That's the paradox. It cuts both ways. And this difficulty in modeling isn't just for complex systemic diseases. Take stroke.
Okay.
Even with standardized protocols like the STARE guidelines, trying to make the animal models better,
Yeah.
we still see failures. There was a drug, NXY059, looked great in animals,
Failed in humans.
completely failed in large human trials. Part of the problem is lab animals usually don't have the other conditions humans often have, like atherosclerosis, which complicate things like stroke.
And other neurological diseases, like ALS.
Yeah, the sources specifically mention ALS, Lou Gehrig's disease. It's widely recognized now that the animal models just don't predict reliably what will help humans.
This seems to be a recurring theme across different disease areas.
It really is. Lots of drugs look promising in animal models, but then numerous big phase 3 human trials failed.
And inflammation.
Even there. Mouse models are used all the time for inflammatory diseases, but research shows their gene responses are significantly different from humans, timing, duration, everything.
Leading to failed trials.
Contributing to the failure of nearly 150 clinical trials for anti-inflammatory drugs, according to one source.
Okay, so rodents, lots of issues. But what about primates? Non-human primates, monkeys. But they're closer to us biologically, right? Does that help?
You'd think so, but even with NHPs, the translation isn't guaranteed. Parkinson's models in primates haven't fully replicated the human disease.
And HIV/AIDS vaccine research, I remember reading that's been a long, difficult road with primates.
Oh, absolutely. Decades of work, huge amounts of money for HIV vaccines.
And the result?
Around 90 vaccines that worked in the animal models failed in humans. 90.
90 different vaccines.
That showed promise in animals, yeah. It raises serious ethical questions, too.
It sounds like even our closest relatives aren't reliable enough proxies.
There are just fundamental differences. The most dramatic example has to be the TGN-1412 trial.
What happened there?
It was a drug trial in the UK. Healthy human volunteers were given the drug. They had catastrophic, life-threatening immune reactions, a cytokine storm.
Oh my God. Had it been tested in animals?
Extensively. Multiple species, including non-human primates, even at doses much higher than what the humans received. And the animals showed basically no ill effects.
None at all.
None reported. It starkly highlighted that even with primates, there could be profound physiological differences.
Okay. So the inefficiencies, the failures, the misleading results, pretty compelling evidence, which leads to the maybe uncomfortable question of scale. How many animals are we actually talking about here?
Globally, the estimate is around 100 million animals used each year for research and testing.
100 million per year.
Yeah, it's a vast number. And that includes mice, rats, dogs, cats, rabbits, fish, birds, monkeys, whole range of species.
And our sources gave a glimpse into the reality for some of these animals. The Macaque Research Center.
Yes, in Puerto Rico. One source included an account from someone who worked there. Hundreds of monkeys euthanized annually, partly for population control, partly for research.
And the description of the process.
It was quite detailed. Euthanasia, dissection. It really brings home the direct impact on the individual animals involved in this process.
It's hard to hear that number, 100 million, and those details, especially when you weigh it against the evidence we just discussed about the low predictive value for humans.
Exactly. It inevitably leads you to ask, are there better ways? Are there alternatives?
And it seems the answer is increasingly yes.
Definitely. Science is moving forward. Our sources talk a lot about the development of human-relevant methods.
Like what specifically?
Things like highly sophisticated assays using human cells, organs on a chip.
Organs on a chip, like miniature human organs.
Sort of, yeah. Tiny microfluidic chips lined with human cells that mimic the structure and function of human organs. Lungs, livers, kidneys, intestines, even multi-organ human-on-a-chip systems.
Wow. And computer modeling, too.
Advanced computer modeling, yes. Using computational power to simulate biological processes and predict drug effects based on human data.
And this isn't just theoretical. Regulators are noticing. You mentioned the FDA Modernization Act 3.0.
Right. That's a significant legislative push in the U.S. It requires the FDA to actually establish clear guidelines for these non-animal test methods to promote their use and acceptance.
That sounds like a big step.
It is. It signals a real shift towards embracing these newer technologies.
And the sources listed specific examples of these alternatives for different body systems. Okay. What about the brain?
For the central nervous system, you've got things like microbrains or brain organoids grown from human stem cells, advanced imaging techniques, EEG monitoring,
And lungs.
in vitro models using human lung cells like the air-liquid interface cultures, and also lung-on-a-chip systems from companies like Emulate.
It seems like there's a chip for everything: gut, liver, kidney.
Pretty much. Intestinal organoids, gut on a chip, liver on a chip, kidney on a chip. Companies are developing these specialized microphysiological systems, Hurotox, microlivers. Lots of innovation happening.
So instead of testing on a whole animal, you test on a specific human-derived system,
Exactly. And you can even use these systems to model specific human diseases.
like Alzheimer's or Parkinson's.
Yes, Alzheimer's, Parkinson's, ALS, cancer. Researchers are creating disease models on these chip platforms using cells from patients or genetically modified cells. It lets you study the disease in a human context.
The potential benefits sound enormous: faster drug development, lower costs.
That's the projection. Some sources suggest these human-relevant systems could potentially have drug development time and cut R&D costs maybe fivefold.
Fivefold. That could mean cheaper drugs eventually.
That's the hope. And importantly, better testing methods mean safer clinical trials for humans
How so?
because you'd have more confidence from the preclinical data. You'd weed out more failures before they get to human testing, reducing the risk of serious side effects or deaths in trials.
That makes sense. And you mentioned earlier that the high cost and risk of the current system might discourage research into treatments for rarer diseases.
Absolutely. If developing any drug costs billions and relies on unpredictable animal models, companies are less likely to invest in treatments for conditions with smaller markets.
So alternatives could open up research for those areas?
Potentially,
Yeah.
yes. Lower costs and better predictability could make developing drugs for rare diseases more feasible.
It's also wild to think that some common drugs we use today might not exist if they'd had to pass modern animal testing requirements.
Right. Aspirin and penicillin are the classic examples often brought up. They can cause problems in some animal species.
And other successful drugs might have been abandoned.
Yeah, drugs like tamoxifen for breast cancer, Gleevec for leukemia, cephalosporin for transplants, even things like Prelosec or Abilify. There were apparently misleading animal results for all of those that could have stopped their development.
It really highlights the opportunity cost, doesn't it? The money and resources spent on animal testing.
Could perhaps be better invested in developing and validating these more predictive human-based methods.
Because ultimately, the human-based methods are based on, well, humans.
Exactly. Rooted in human biology. It just removes so much of the guesswork involved in trying to translate findings from a mouse or a dog or even a monkey to a person.
Okay, so let's try to sum this up. We started with the history, seeing how animal use in research evolved over centuries, but only became formally required relatively recently.
Right. Then we saw compelling evidence about the inefficiencies, the high rate of adverse reactions missed by animal tests, the staggering 95-96% failure rate of drugs in human trials.
Basically, animal tests being no better than chance at predicting human outcomes. And we looked at specific examples where animal models just failed to replicate complex human diseases like stroke, cancer, Alzheimer's, HIV.
Yeah, even with primates, the translation is unreliable, highlighted by cases like TGN 1412. Then the sheer scale, 100 million animals used globally each year.
And finally, the really promising developments in human-relevant alternatives. Organs on a chip, advanced cell cultures, computer modeling, methods based directly on human biology.
Methods that offer the potential for faster, cheaper, safer, and more effective drug development.
Which leaves you, the listener, with a pretty significant thought, doesn't it?
Yeah. Given all this data, the historical context, the documented limitations of animal testing, and the rise of these sophisticated alternatives, should we reconsider this long-standing reliance?
Is it time to shift focus towards methods that are potentially more effective, more efficient, and, well, avoid the ethical concerns surrounding animal use?
It's a critical question for the future of medicine, impacting both human health and animal welfare.
Definitely something to think about. We'd encourage you to look more into this, maybe research the FDA Modernization Act 3.0, see what these alternative technologies like organs on a chip are all about.
The field is changing fast, and understanding these shifts is really important.